A colleague and I developed a novel therapeutic to induce apoptosis of tumor cells. And we had actually initially developed this protein therapeutic and tried it in vitro against tumor cancer cells. And we found that in cell dishes, it actually was able to induce cell death pretty dramatically, a variety of types of tumors. And so we were quite enthusiastic about this, but when we tried to apply for funding, it was a little bit difficult because everybody really wanted to know whether this worked in vivo. And so what we really needed to do was find the funds in order to do testing in an actual animal model. So given that funding is difficult these days, and we were having a hard time getting actual interest in funding it at this level, though there was a lot of interest in our concept. So we were actually directed to the Catalyst program by Bill Seaman, who is the Vice Chair for Research in the Department of Medicine, who had heard about this program and suggested we apply for it. When we did, we actually didn't really understand what we were getting involved with. It said we were going to have a consultation, and we didn't really know what that meant. And we had a variety of consultations with a variety of senior investigators involved in both, that were very experienced in both IP issues and in protein production and in translating real novel therapeutics into treatments. And so with their advice and with some funding we got from the Catalyst program itself, we were able to do a pilot study which enabled us to study our novel therapeutic in an animal model against a renal cell carcinoma. And pretty dramatically, in a short study, we were able to show that our protein did indeed induce apoptosis of the tumor. In other words, it killed the tumor cells quite dramatically in a way that was quite novel.